Good morning. I'm not Walter Wynn. Walter is off to the ophthalmologist taking his wife for an appointment this morning. So he asked me to fill in for him, and I'm pleased to do so. It's my pleasure this morning to introduce Rachel Newsome, who is a graduate student in the Cancer Center at Shands. Rachel was an undergraduate here at the University of Florida in the Department of Chemistry, where she worked with bacteria, studying very mysterious genes and their products, which I can't pronounce, so I'll let it go. But anyway, after she graduated, she worked for 2 years in Dr. Jobin's laboratory as a technician. That was so successful that she joined the graduate program in biomedical sciences. Let me say that somebody early on in the history of College of Medicine was a genius, because we set up a graduate program called the Program in Medical Sciences. That covers anything that anybody wants to do in the health center. It doesn't have to go through all of the bureaucracy if you want to do something slightly different. And that's worked out incredibly well over the years. And one of the real beneficiaries has been our cancer center. So Rachel's still working with Dr. Jobin. She's gotten interested in the microbiome. We all know that we are 90% bacterial cells and 10% human cells. And obviously that influences our health and well-being. And Rachel's going to give us some details on that situation. So welcome, Rachel.
Thank you. Thank you, Dr. Burns. That was a very kind introduction. So I'm glad we have a small crowd here today, because it'll be a little more interactive. So I have to ask this question, which is, how many of you are affected or know someone who has been affected by cancer? Literally everyone. Cancer is a disease of the elderly, unfortunately, so to be expected. But what we do in the lab is we look at the connection between your microbiome and your immune system and cancer. And we look at how cancer is developed and then how we can treat it. So some of you may have been to a presentation by Dr. Jobin. So he is my advisor. He's a rock star in name only. He does not play any musical instruments. But we have a huge group at the College of Medicine. And like Dr. Burns said, almost all of us are in the biomedical sciences program, which was started many years ago. And it allows us to do pretty much anything we want within the College of Medicine with a very defined course load, and it's really interdisciplinary. So it's been a huge benefit to my graduate career being part of that. So the outline today, we're just going to discuss the background of the microbiome and immunity and how that relates to you. We'll talk about some various diseases the microbiome's influenced by and influences. And then we'll talk about cancer, which is kind of my passion. And we'll talk about a little bit about my project and the company that Christian and I founded that we're just getting off the ground now. So as Dr. Burns said, bacteria, their cells outnumber our cells, which is incredible to think about. And this was largely ignored until about 10, 15 years ago, except in the cases of pathogens like E. coli or C. diff. And so now we're really interested in seeing, what makes these microbes so important to our health. And one of the reasons why they're so interesting is their gene content. So when you think about our human cells, and you do have to consider external factors such as splicing differences, which does increase the number of genes we can make, but bacteria have far more genes than we do. And the vast majority of these are undefined. So we don't know what they produce, we don't know what they do. And a lot of the bacteria can actually share these genes back and forth, which adds another layer of complexity. So what we're really interested in seeing is defining these genes and how they interact with our host system. So the intestinal microbiome contributes to host health and disease. And this is done in a variety of factors. So many factors actually influence our microbiome. So if you have inflammation in your gut through a genetic mechanism such as Crohn's disease or ulcerative colitis, that changes your microbiome. Other things such as diet and lifestyle, exercise recently has been a huge factor. We're actually looking at that in the lab right now. Long distance runners have a totally different microbiome than people who lead a sedentary lifestyle. And some of the reasons people are interested in that is, can you do a fecal transplant from a marathon runner into someone like me, and then is that going to make me a marathon runner? I don't think so. But we're looking at, can we help people using these factors? And so when these factors influence the microbiome, this in turn tips the balance between health and disease. And so when we have a shift in the microbiome from your normal, and what your normal is based solely on you and your environment, when we have a shift in that, we call that dysbiosis. And so it's a substantial shift just in what your microbiome looks like. And so this can help with health. So if you don't have dysbiosis, what the microbiome generally does, it helps in nutrient acquisition. So your microbiome digests a whole lot more than you're able to digest. And additionally, it can exclude pathogens such as C. diff and E. coli. It's actually able to naturally work around these things and exclude them. But then if you have a dysbiotic microbiome, these things can shift the balance towards other disease states. So this includes obesity, diabetes, also autoimmune diseases. And so these are multifactorial diseases. I am not saying that the microbiome causes any of these or that these diseases cause a shift in the microbiome, but there are features that are shared between these diseases with dysbiotic microbiome, and people are still studying that relationship. So some of the strongest signatures are IBD. So if you have colitis, ulcerative colitis, IBS, these are diseases that we actually do think have a causative factor with the microbiome. So if you have a little bit of inflammation, then you may get more dysbiosis, and that in turn will lead and snowball into a worse disease. But some other ones which are really interesting include rheumatoid arthritis and MS. And so these actually have a defined signature, but how this actually happens, people are still working on this. This is the problem for people much smarter than me. Many of you know that there is a direct link between bacteria and cancer. And this is kind of defined mostly in bacteria that we're familiar with now. So H. pylori and gastric cancer, for example, and then some viruses like HPV, and we have vaccines for those now. HPV does cause cancer. I think it's ovarian cancer. I think you guys probably know. Cervical cancer, yeah, there we go. See, I'm not a virologist here. So there are many connections, direct connections that have already been established. But what we're looking to do is establish the connections that are a little more tenuous. So we look at how the healthy microbiome and the disease microbiome produce metabolites that differentially regulate the immune system. So how we theorize this happens is that the healthy microbiome produces these secondary products, either through digestion or interaction with other microbes. This in turn influences the immune system and causes normal homeostasis. And then our theory is that when this is dysregulated, then that's when you get cancer. And so some of the bacteria we look at are E. coli that actually contain genotoxin products. And it's very interesting because even though we've shown in mice that these genotoxins do cause cancer, in humans, the connection is a little bit more nebulous. We're not exactly able to prove that all people that have colorectal cancer have this bacteria. And all the people that have this bacteria don't necessarily develop colorectal cancer. So it's not a hard and fast rule. So what makes a healthy microbiome? So I mentioned this thing called diversity. So diversity is the richness of the bacteria in your gut. And generally people say that better microbiome is more diverse. And that's not necessarily true. So don't quote me on that. It depends on the context for sure. But in our lab, we profiled every single member of our lab by collecting feces from everyone. And we use this technique called sequencing, 16S sequencing. So we take the bacterial ribosome gene and we cut a small portion of that and we actually barcode it and sequence it with next generation sequencing. It allows us to completely profile the microbiome of every single person who donates a fecal sample, and we're able to relate the composition of each person to another by condensing all of those factors into a small data point, and that's done by bioinformaticians, not me. I've tried my hand at it. It's very, very difficult. So it's a lot of computer work and programming. But what that generates is these things called PCOAs. And so this is a PCOA of our lab in 2021. Christian was very proud because he was the most diverse microbiome out of everyone. I think he was 111 or something. And then we had a lab member who recently took antibiotics. I think he was 83, not to out him. So less diverse is not as good, but context dependent. I was number 9 when I was really, really depressed because I ate yogurt for 6 months leading up to this test, try to beat everyone else, and it didn't work. So, so it really, just eating yogurt every day is not going to solve all your problems. But the people who have the less diverse, their gut microbiome kind of looks a little bit like a cornfield that only has corn. So it's not very diverse. There may be a lot of bacteria, but it's of just a couple types. Whereas the people on the left-hand side, they look more like a rainforest with thousands of species. And so if you think about it, if you have an invasion of maybe kudzu or something invasive in Florida, it's going to be a lot more effective in the cornfield than it will be in a rainforest. I mean, of course, it depends on temperature and climate, as you biologists in here probably know, but you get the metaphor. So what we look to do in the lab is how do we impact the health of the microbiome, not just through diversity, but through other things. So currently, there are clinical interventions that use fecal microbiome transplant and pre-probiotics diet to influence the microbiome. These are done on a daily basis, not for everything and not specifically for cancer, but that is starting. But what we look at and what we're more interested in is these more targeted therapies. So we don't have to give you an antibiotic treatment followed by an enema, followed by a huge bag of fecal transplant. If we can just give you, let's say, a bacterial metabolite that does the same thing as if we gave you a fecal transplant. So this is kind of where the field is going now. So we look a lot into translational research in the lab. So some of you are probably familiar with the translational research spectrum. So many of these bacterial metabolite studies are still preclinical. They have not even entered into the realm of thinking about an IND. But when you talk about fecal transplant, a lot of these are in phase 1 and 2. So phase 1 clinical trial is safety as the primary endpoint. You only look at efficacy if, maybe somehow secondary, you see something, but primarily it's for safety. So this can be done in healthy patients. In cancer trials, we generally use cancer patients because they need to be undergoing therapy at all times. But we look for adverse events primarily, first and foremost, before we look at efficacy. And so if your therapy is safe, so if your fecal transplant or something is safe, then you can move to phase 2. This is a lot more expensive. It's a lot harder. The primary outcome is efficacy, and you need to enroll a lot more people. And then if phase 1 and phase 2 work out fine, then you can move to phase 3, which is almost impossible to complete. Many drugs never make it past phase 1. Most drugs don't make it past phase 2. And maybe 1 or 2 drugs out of the hundreds and hundreds that are tested make it phase 3. And it takes years. And if you complete phase 3, which is multi-center, so you need to have many centers across the country, diverse population, if you still get efficacy as compared to the standard of care therapy, then it will be approved by the FDA. And then you will start the implementation. So when you actually put this into the clinic everywhere as the standard of care, do you still see that there's an effect in the population? And a lot of times you'll pass phase 3 with a trial, and then you'll see that in real life, it doesn't work any better than any other therapy, which is kind of sad. So what this creates is kind of a cycle. So you have therapies that are starting phase 1, phase 2, moving to phase 3 and 4 that are failing, and then moving back to phase 1 to see what went wrong, how can we improve it. So we're focusing on phase 1 and 2 in the lab and at the cancer center. And now that we have NCI designation, now we'll start to be part of the multi-center trials that are so coveted by everyone in the cancer field. And I already kind of discussed that. But what we're looking at with fecal transplant specifically is, how can we maximize the potential when we're in a clinic and in a cancer clinic? So the fecal transplant that we do in the lab is into mice mostly, but this is done in the clinic and it is done in the context of cancer. So what they do is you take donor feces, and these are from healthy people. And when they say healthy, they're talking like between the age of 18 and 30, so young, and then people who have not been on antibiotics pretty much ever, and people who, have a great BMI and they're active and no other secondary comorbidities. So you can pretty much take it from any person who meets those guidelines. And what they do is they literally put it into a blender. So I've seen this done in the clinic. You just take all the feces from one patient, put it in a gross blender in a gross GMP facility, and you just blend it up. And anyone can really do this, but I would not suggest it. Anyone can do this process, and then you essentially filter it and then give it to the patient as an enema. So it's really nasty. Do any of you watch South Park? South Park? Yeah, have you seen the episode with the feces where they take Tom Brady's poop because he's like the pinnacle of health and wellness, and everyone's trying to steal his poop by siphoning off the pipes in his bathroom? It is pretty funny. But that's not too far off from the truth. If you could take the feces of Tom Brady and blend it, you could give it to someone, but you need to go through a pretty rigorous safety trial to make sure that you're not transferring any communicable diseases from these patients, et cetera. So when I say donor selection can be any healthy person, it really does work. And so for C. difficile infection, which is a common enteric pathogen, causes diarrhea, if you get a fecal transplant with recurrent C. diff, it's much more effective than antibiotic therapy. And so there was a famous study that was in the New England Journal of Medicine where 5 donors, 78% of patients without any routine screening, they just had recurrent C. diff, were able to overcome their recurrent infection. So the regulations have to be pretty structured when it comes to these patients. Like I said, the filtering and the homogenization with the blender is not so complicated, but you really need to be sterile. And handling is a little bit complicated because the majority of the bacteria in your gut are anaerobic. So they cannot live with much oxygen. So when you actually poop out the bacteria, they're immediately exposed to oxygen. So you have to move very quickly. You have to use reducing media in order to make sure these bacteria are viable when you're actually transferring them. And then additionally, individual stool pathology. Have you all seen the Bristol stool scale? No? So it's like a scale of 1 to 8. The 1 is like little hard rabbit pellets, and 8 is like pure liquid. So you really want something that's kind of in the middle. Nice log, not too PC, but not too liquidy. So that's what I mean by individual stool pathology. And so there are companies that actually will pay you for your poop if you're interested. You have to be local to the Boston area, but if you're looking to make a little cash and you're healthy, you can actually donate your feces every month to a stool bank called Open Bio. And they homogenize it and they freeze it down, and then clinics across the world can actually buy this feces and use it to give to a patient. So you can actually be helping the world and making a little money on the side. It's pretty cool. But we're in Gainesville, so. So a couple of famous studies have been conducted using these fecal transplants. So at Stanford, Kathy Nagler, who gave a talk just a year ago at our cancer center, she did this twin study. So they took twins who were discordant for peanut allergies. So one twin, identical twin, so genetically identical, had a peanut allergy, and the other one didn't. And so they characterized their microbiome and found that they were different in one factor, which was this Clostridia species, Rheumnococcus bromi. So it forms spores, and twins who do not have the peanut allergy have this, and the twins that have the peanut allergy do not. So they've actually started a company where they target these Clostridial species using diet interrogation and all sorts of things. And if you feed potato starch, you can kind of increase the abundance of this bacteria. So they're really trying to use the diet, use the microbiome in order to improve peanut allergies. And this is in children. And so this is the company they have now. And so instead of actually giving the microbe to these patients, the Rheumnococcus, they're trying to drug it through the diet. What they're actually doing is they have a backbone that has the metabolite produced by the Clostridial species. Then you don't even have to worry about the fecal transplant or anything like that. So that's just a little background on the microbiome, disease, health, etc. So now we're interested in cancer, right? We're interested in disease detection. We're interested in all of these things. So early detection for cancer is key. The earlier you get it, the smaller the tumor, the easier it is to treat. As soon as it becomes metastatic, the treatment complexity goes tenfold higher. You have to do radiation and chemotherapy at the same time. Targeted interventions don't work as well. So early detection is a really important part of cancer therapy, even though it's not a therapy itself. And so this new cancer screening method has come through. So in general, in the clinic, when you are getting biopsy for cancer, they're either going to do a needle biopsy. So that's going to target one specific site. It's going to take a little piece of your tumor. May not be representative of all your tumors or all types of cancer that are in that single tumor. So it's really limited when it comes to that. Or you can do a body fluid marker, so PSA. This may not be specific to the cancer. It's a secondary readout, so it's not as good. And you can also look at other biomarkers. And of course, imaging is the gold standard, but how many times are you really going to go get an MRI? And you're exposed to radiation every time you get imaged. So what we're interested in the lab and looking at is liquid biopsies. So this is where you're able to take a blood sample and then use detection techniques on that blood sample. So many of you probably heard of Theranos, the detection company. Elizabeth Holmes, yeah, boo, boo. She said she could do this with just a little pinprick of blood, which is crazy. So in the clinic right now, you need to give like a liter of blood for these tests. That's where the cell-free DNA, microbial DNA, circulating liquid biopsies are kind of derived from right now. So we're not talking about the Theranos side. We're talking about the real diagnostics here, and it's very difficult. But what we look at in the clinic is circulating tumor DNA. So your tumor, as it grows unchecked, basically, but a lot of these cells will die through apoptosis, or they'll have a mutation that causes them to be unviable. So your tumors do shed DNA, and they shed them at a pretty high rate, and they shed them throughout the body in the circulation. And so what we're looking at is, can we sample the cell-free tumor DNA? Is it representative of someone who may have cancer, what type of cancer it is? And then can we use the microbiome signature from circulating tumor DNA to actually examine what type of cancer you may have? So how it's done currently is you get screened and then you can monitor a response. And then we use quantitative analysis and genomic analysis of these samples to look at, what are the tumor mutations we're seeing? And you can also look at the microbiome in these samples. So these need to be personalized to the patient. That's why Theranos was so unsuccessful. And they weren't just looking at cancer, of course, but when it comes to diagnostics, you have to think about the exact mutations a person has and how the tumor sample may be different from those. This is really something that needs to be specific to every patient. So what we've kind of designed is for non-small cell lung cancer, we look at sampling, we do whole exome sequencing. This is whole DNA sequencing of the whole body. This would be representative of pretty much every, all the DNA that you have. And then we look at phylogenetic analysis to see what's normal in your body, what's not. And then we can take a blood sample and then look at pre-surgery and relapse and monitor, how your tumor is doing based on your baseline. And so this kind of provides a little bit of a holistic approach. And so our contributions to this as microbiologists is that in addition to looking at the DNA of your body, like I said, we're also looking at the bacteria that may be shed and present in the circulation. And so these are generally not alive. They're dead bacteria and dead DNA that's just circulating. And so when they sample your blood in the clinic for something like this, then we also look at the bacteria that are present. And it turns out, if you look at specific tumor types, different tumors harbor different bacteria. And we're not really sure why this would be. So colorectal cancer has one specific type of bacteria, brain cancer has another, and we don't really know how to explain that or why bacteria would co-localize with the tumor. But what we're able to do is make those connections and then combine that in a machine learning pipeline. And we're trying to use these in the clinic now to see if we can monitor where cancers are located based on the bacteria that are in these samples. And so that's boring stuff. But this is the most exciting. So cancer therapeutics is probably the most important part of the research that we do in the lab. So traditional therapies like chemotherapy or radiation for cancer have been used for decades, but recently cancer immunotherapy has been the biggest kind of up and coming treatment in the past several years. Several types of immunotherapy include immune checkpoint inhibitors. You can have adoptive T-cell therapy, CAR T-cell therapy. All of these really harness the patient's immune system to fight their own cancer. And so it turns out that the microbiome can actually influence your response to immunotherapy. And the way this happens, we're not really totally sure, but specific types of bacteria are present in people who respond to these therapies versus those who are not. And we think it's an interplay with the immune system that's happening. And this is also present in other types of therapy. So like I said, chemotherapy, radiation, the gut microbiome actually plays a part in those as well. So we know this is because the microbiome influences drugs. It can degrade them. It can enhance their effect. For example, irinotecan is a chemotherapy. It's a prodrug. A lot of people get it. And it turns out that the microbes can convert the inactive form to the active form. And this happens in the gut. So when your body is trying to metabolize and get rid of the excess irinotecan after it's done killing your tumor, your bacteria can actually reactivate it and then destroy the lining of your gut through active chemotherapy. So this is one example of that happening. Another for Parkinson's disease, the microbiota can actually metabolize L-DOPA, the drug that's used to treat Parkinson's disease. It can metabolize it in the periphery. So they found in the clinic certain people responded to certain doses of L-DOPA for no reason, and other people needed way higher doses. And it was because certain people had this bacteria that were digesting the L-DOPA. And so lastly, gemcitabine is a chemotherapy, and bacteria in pancreatic tumors can actually deactivate gemcitabine. They can metabolize it. And so even though you're getting chemotherapy for your pancreatic cancer, if you have this bacteria in your tumor, it's going to degrade it and it'll be less effective. So there are many examples of this happening already, but what I'm really interested in is in immune checkpoint inhibition. So this is not chemotherapy or radiation, this is immunotherapy. And what it does is naturally your T cells have this signaling pathway. So it's called PD-1, PD-L1. Your body expresses PD-L1 all the time. All of your cells express it. But when tumor cells are growing, they overexpress PD-L1. And as a result, when your T cells detect a mutation in your tumor cell, but they also detect PD-L1 expression, they will turn off. But we use a monoclonal antibody. It's given IV to patients. And when we give them this, it will interrupt this signaling pathway and your T cell will be able to kill your cancer cell, which theoretically should work great, you would think. But actually, a lot of patients don't respond well. And a lot of cancers don't respond well, particularly solid tumors. So have any of you heard of these drugs? Keytruda, Opdivo, Yervoy, they're pretty common now. These are all immune checkpoint inhibitors. Some of them target the pathway I was talking about. Some of them target others, but it's a huge market now. So people are really trying to figure out, how can we enhance response to these immunotherapies? So what I did as part of my PhD was, in a collaboration with Moffitt Cancer Center over in Tampa, we profiled the gut microbiome of patients who responded and didn't respond to immunotherapy, who had non-small cell lung cancer. And they had late stage non-small cell. So this is metastatic disease, which is what we would use this for in general. And so about 20% of our patients did not respond, did respond, and the vast majority did not. And so we were interested in seeing, why is this happening? Also, signal-wise, 2 studies that had been done previously on other cohorts in other countries found the same bacteria was enriched in responders and enriched in non-responders. So there's a signal problem. We're not able to really say what bacteria is causing the response. And so from our cohort, which was done in Florida, we took the fecal samples. We found that they were different between responders and non-responders. We took the fecal samples, and what I did was pooled these into mice. And so I gave them a fecal transplant, and I used a mouse model of cancer. It's not specifically lung cancer. It's called Lewis lung carcinoma. Famously does not respond to pretty much any therapy. I gave them a fecal transplant, I gave them lung cancer, and then I treated them with immunotherapy. So our immunotherapy is not under a drug name, it's just anti-PD-1, but it's just a monoclonal antibody that inhibits that interaction. And what we saw was our responder donor mice recapitulated the phenotype of their donor humans. So we were really interested in seeing why this may be. We had profiled the microbiome of our patients and our mice, and we found that Bacteroides, this is a genus of bacteria, was equally enriched between responders and non-responders. And so this is at the genus level, and with sequencing, you can really only get to the genus, maybe species level. So we were really interested in seeing, what is different from the bacteria that are enriched in the non-responders versus those that are enriched in the responders. I checked the tumors also to see if there's a presence of a microbiome, but there really wasn't. So we kind of ixnayed that direction. We went directly to the responder feces. So we have this platform in the lab where we're able to anaerobically grow isolates from human feces. And then we make a library of these isolates, and then we identify them. We're able to pull them down and use them for experiments. So we identified about 700 isolates from our responder feces from these patients. And what we were interested in seeing was, of the Bacteroides isolates, there are 183, what makes them different and what makes them respond or not respond? And so we actually screen these isolates for immune activation, and we screen them all individually. So all 183, we screen them through a process, just immune activation. I'm sure many of you are familiar with CD8 T cells, but I don't want to get too technical because it's boring. But we found that of the 183 isolates we screened, 6 were stimulatory, which is really exciting. So 6 of these isolates we found produced this phenotype that would be anti-tumor in a person. So we took these 6 together and we gave them to our mice, and we compared them to our responder feces and non-responder feces given PD-1 therapy. And we found the same thing. The 6-member consortium actually recapitulated the response phenotype of the responder feces. So now we know there's something present in the responder feces, also present in these 6 bacteria, that's promoting response. And so we characterized the tumors of these mice. We found they were more immunogenic, more anti-tumor, CD8 T cells. And so what we wanted to see then is, what are these bacteria producing that could be causing this? Because like I said, giving live bacteria to a patient, it's a little bit difficult, especially in a clinical trial, especially submitting an IND. So we took these bacteria and we actually screened them for small molecules. What we found was that small molecules produced by the consortium caused this effect. So what we then did was this bioassay guided fractionation, and we did this in collaboration with Yale, which is a big name, but UF is better than Yale. UF is the best. And so in collaboration with Yale, we took a single bacteria from these stimulating isolates, and we performed bioassay guided fractionation. All this does is we took all the products produced by a bacteria, did 6 crude fractionations, so 6 by size, essentially. We fractionated the supernatant 6 times. We screened each of those 6, found only 1 fraction produced the effect we had seen previously, took that fraction, and then further fractionated that into 12 more fractions. And this is, again, crudely by size. And then we screened all those 12, and we found only 1 fraction produced the effect. So now we know that there is a small molecule or several in this fraction that's responsible for the bacteria's effect. So after large-scale culture and tandem mass spec, if some of you are chemists, I am not. I used to be, but now it's way out of my wheelhouse. I haven't done NMR in a really long time. But if you take small molecules, put them through mass spec, you're going to see somewhat of their structure. And then we used some computational pipelines, and we found 5 small molecules that are present in this fraction 11, the stimulatory fraction, that are present nowhere else, not in the media, not any other fractions produced by the bacteria. And so it turns out the structure is pretty simple. It's just a small molecule with a long lipid tail. We call it BAC430. And when we took this small molecule and we did dose response, we found that the cis version of BAC430 produced a significant dose response, especially at kind of lower concentrations, whereas the trans version did not. And it also promoted a little bit of increased viability in our cells. So we were interested in seeing, if we take this compound, if we give it to mice, what's going to happen? Are they going to respond to immunotherapy better? And this is without any changes in the bacteria at all. So we did intratumoral injection of this small molecule, and we found the mice that received the small molecule had significantly better response to immunotherapy. And this was with the same lung cancer model that doesn't respond to anything. So now we formed this company. It's called BB Therapeutics. It's short for Bébite, which means little critters in French, which is Christian's nationality. And so now what we're looking at is how can we optimize delivery. We're starting an IND application, so we'd like to bring this to a clinical trial at UF, just for safety at first in non-small cell lung cancer patients. And then another platform of the company is characterizing other small molecules, both from this consortium and then other bacteria, seeing if we can develop those into drugs for cancer treatment. And so that's kind of the research that I did as part of my PhD, and that's coming to an end now. But that's all part of the lab of Dr. Christian Jobin, collaboration with Yale, and of course, a ton of funding from the Cancer Center and the clinical and translational research building here at UF. And then I'd like to take any questions you guys may have or have a discussion about the microbiome and cancer. Yeah. You first? Oh, yeah. Yeah.
What's better? There was a 62-week plate. Is that the best?
No,
Rachel, can you repeat that just for the Zoomers?
Oh, of course. So what he's asking is, when it comes to PCOA, does location matter? Is that better? And so when you're saying, okay, so better is not always diversity related. So the PCOA only shows the differences between people. And so the color coding actually shows the diversity level. So what's really interesting is the people that were really diverse, gotta go way back. Oh yeah, here. The people that are really diverse kind of cluster together. So I would say the people on the left-hand side are better. The people on the right, this is a newborn, 62, so really, really not diverse microbiome. So to the right is a little bit worse, but it really depends every time on the group of samples you have. The PCOA doesn't really tell you who's healthy and who's not. It just tells you who's different.
Thank you for all the scoop. And I wonder if you could just summarize where this all is in terms of applying it to, say, ovarian cancer.
Exactly. So actually, ovarian cancer, there is a strong connection between the microbiome and treatment response, not so much development. So with patients who have these solid tumors like ovarian cancer, non-small cell lung cancer, what they're doing now in the clinic is fecal transplant from healthy donors as an adjuvant therapy, followed by immune checkpoint inhibition. So that's what's currently being looked at. There are no small molecules in clinical trials that are from bacteria. There are no therapies that would do intratumoral injection of bacteria or products to ovarian cancer, but I think the next generation of therapies are going to be those targeted therapies. So it's either going to be bacteria that are being injected into the tumor to cause killing, or it's going to be bacteria that are given as a probiotic to patients who then receive immunotherapy.
So it sounds like it's in clinical trials.
Yeah, nothing has been approved as far as ovarian cancer, and it's an incredibly difficult cancer to treat. It's very similar to pancreatic cancer and glioblastoma in that it's very invasive response to very few treatments. So that's going to be one of the last ones that we probably gets approved for.
And finally, any estimate, it sounds like that's quite down the road for that particular ovarian cancer.
Yeah, probably 10 years.
Well, that's not bad.
Which is not bad. No, it's not bad. I mean, cancer therapy has come a long way in the last 10 years if you think about what people were getting 10 years ago. So yeah, we may be close. I don't think we're close to a cure, like a pan cure for cancer at any rate. But, I think we're going to see progression-free survival increase way beyond what we've seen.
Well, thank you, Rachel.
Thank you.
Thank you, Rick. I see your hand raised. Go ahead.
Yeah, thank you so much for the presentation. Could you tell us about how you organize the company for the small molecules, how UF supported you, and whether there's any patents involved?
Thank you, Rick. So Rick asked, how is UF involved in the development of the company? Are there any patents involved? And I can say for sure, UF has been crucial in the development of this company, right? So all of the grants that Christian gets have to go through UF, and so they give us, our disbursement funding, which funded all of this research. And so when we thought we had a patentable drug, the small molecule, we actually went to the UF Innovate. So they have the Office of Technology Licensing. And so we went to them and we said, hey, we think we have something that could be patentable. What do you think? And they said, yeah, definitely. So UF, I don't know what the terminology is. They prosecute the patent or they file the patent. So UF has done all of that for us. They have a law firm that formatted the patent. Basically, we just had to submit data to them. They took everything together, filed the patent. So UF owns this IP on the small molecule and also the bacteria. And so when we decided to form the company, which was way after we had this patent, we went to the Office of Tech Licensing. We said, we want to maybe form a company based on this. And they told us, okay, you are going to have to license an option agreement from us for your patent. And this can be an exclusive license agreement, which is wonderful. So it means that no other company can use this technology. And how it's going to work is UF is going to take 5% equity of the company as a payment for the licensing of our technology. And that maybe down the road, like 5 or 10 years, if there's enough equity in the company and it's valuable enough, UF is going to sell that. And then they're going to retain the profits from selling that stock and that equity as payment for licensing the technology. And then we'll be able to own the patent. So that's kind of how it works so far. We recently incorporated, so we have three members of the company right now. We have our CEO, which is me. Christian is our president of the board of directors, and we have a CFO. And that is it. So it's a very small company right now. And also UF is helping us submit SBIR grants. These are grants from the government that fund small businesses that are related to health and wellness. And so we've applied for funding for our small business that will be non-dilutive. So they won't take equity for this funding. Venture capitalists are not going to own a portion of the company for this funding. We can just use it to build the company and build the product without losing any equity. So it's been really fantastic working with UF. They've been incredibly helpful, in the whole process, especially for someone who has no idea about business or finance. But I'm learned what equity was a couple weeks ago. So it's been a really fun journey. And I know many of you probably have started companies or been through that process. So I'd love to talk to you more, if you have any feedback on what it's like getting it off the ground.
Thank you. Any other questions or comments?
Oh, yeah, this is actually very interesting. So,
Can you repeat what he said?
oh, yeah. So Dr. Burns said, can you talk about the conflict of interest? And this is really important because our company is doing commercial endeavors that, of course, should not be funded by grants the lab is receiving to do research at UF. So that's number one. Then number two, our company would like to initiate a clinical trial, of course, based on this therapy. And because UF is now an NCI designated cancer center, we want to go ahead and do that at UF, take advantage of all these great resources we have. However, the conflict of interest there is that they do not want investigators running trials at their site of discovery. And that's for many reasons. It protects against misconduct by scientists, physicians who may know what the goal of the study is and then influence the outcomes somehow, either through grading of, radiology or something else. So there is the possibility there for bias, right? And so for the conflict of interest for UF and the company, really we've been navigating that by making sure that everything that's done as part of the company is completely independent and discrete from the lab. So we have a separate space for the company. At the Innovation Hub, we only use materials that are provided by the Innovation Hub to do that work. And then secondarily, we're talking with the cancer center directors now about how to navigate the conflict of interest for our future clinical trial. So what we have to make sure is we're able to say that the trial has to be done at UF because of X, Y, and Z. So for us, it's because we need to process the fecal samples immediately upon collection. And we are the only lab that could do that processing realistically with scientific accuracy for this small molecule. So we need the location to be at UF. And that's how we're kind of going to circumvent it in the future. But there have been other trials that have had this issue. They've had to go to other cancer sites, such as Moffitt or the Sylvester Miami Institute. So something we're still working through.
Any other questions? Rachel, I know on behalf of Walter Wynn, we thank you for your research and your time and sharing with us today.
Oh,
Thank
thank
you.
you so much for having me. It was an absolute pleasure.